Hi all, I'm here to talk to you guys through some cell Molec fundamentals that I hope you'll be taking with you as we start this course. These are things that you hopefully should have learned in any fundamentals of molecular biology course that you took, but it may have been a while for some of you since you've taken that, so let's get right to it. These are some of the main elements of life that you will see throughout this course in all of our macromolecules. You've got your hydrogen, your carbon, oxygen, nitrogen, phosphorus, sulfur. Right? These guys are the main components of all of your cells. You should know that your organic compounds are going to contain carbon by virtue of the fact that it can form four covalent bonds with any other atom, which makes it a really good building block, right? So we're going to have molecules that have what we call a carbon skeleton, where the central structure of it will be made up of carbon. You'll see this in all kinds of shapes, straight, branched, ring, etc. But the four main groups of these macromolecules that we'll go through, some in more detail than others, are your carbohydrates, your lipids, your proteins, and your nucleic acids. And typically what you'll see is some kind of monomeric unit that is going to link together to make a polymer for these larger macromolecules. But let's think first about how atoms interact with each other. There are two main kinds of bonds that they make. They either make covalent bonds or they perform non-covalent interactions. In a covalent bond, the electrons are shared. This means that each atom is holding on to these electrons really tightly. It means that they're not coming apart from each other. Think about like two dogs going after the same bone, or if you've ever interacted with a dog who's trying to play fetch with you and they're not an especially well trained dog, like my brother's dog, he'll bring the ball to you and then he won't let go. My brother's dog has a covalent bond in one sense with the ball. But these bonds can be single bonds or double bonds. Typically, once in a blue moon, you'll see a triple bond, but that's pretty rare. And we can say that even within the covalent bonds, that this bond is polar, in which one atom is more electron greedy, and that electron spends more time with the atom. And that is to say, you get almost a partial charge, in quotation marks, so to speak. It's a polar bond. Or you could get a nonpolar bond, where the covalent bond does not have any kind of this unequal sharing. And in better terms, right, the electrons are equally shared. So you don't get any of this kind of fake charge business going on. All of our covalent bonds are really strong. These atoms are not letting go of their shared electrons. Okay? And so you'll see covalent bonds in any kind of structure of the macromolecule, some post-translational modifications, actually all of them, I think. You'll see them in histone modifications. We'll get to the histone modifications later on in this unit. On the other hand, in a non-covalent interaction, your atoms are not sharing electrons, right? They're not holding on to each other really tightly. These non-covalent interactions fall into two categories. They can be hydrophilic interactions, where you have this kind of charge element going on, whether it's an ionic bond, where the atoms have outright charges on them, and you have one positively charged atom interacting with a negatively charged atom, making this strong ionic bond, still nowhere near as strong as a covalent bond. Or you could have a hydrogen bond, where you have hydrogens interacting with oxygens, and I'll point out a few examples of this as we go throughout this mini lecture. You can also have hydrophobic interactions, where you have long stretches of nonpolar regions, or they could also be considered regions that don't want to interact with water. Water is a polar substance. Polar and nonpolar don't generally mix. So any kind of large swath of nonpolar region of a protein isn't going to want to touch water. It's going to want to touch other hydrophobic regions, other non-polar regions. And so that will form this non-covalent interaction that will bring those parts together. Non-covalent interactions are very weak by themselves, but if you have enough of them all kind of grouped together, you could have a strong interaction there. And so non-covalent interactions are typically what we think of when we say that one protein is binding to another. They're coming together, they're interacting in a very specific way, but they're not covalently bonded such that the protein can split off from that protein that it's been bound to. So again, you have your protein-protein interactions. You have a lot of non-covalent interactions for secondary structure. Think about all your proteins, the alpha helices, beta sheets. Those are lots of hydrogen bonds. The bringing together of the two strands of DNA to make the double helix, that's all hydrogen bonds with the nitrogenous spaces there, or any kinds of hydrophobic effects that drive protein folding. We'll talk more about this later in the course. But here's, again, another look at your covalent bonds. Your electrons are shared between atoms, right? So this carbon has 4 electrons all hanging out there. 4 electrons. I'm trying to do this laser pointer thing, and it's not. There we go. Your hydrogens also have individual electrons. Electrons don't like to exist on their own. They like to exist in pairs. And so now the carbon and the hydrogen have brought their electron to the table. They're sharing it. They've made this covalent bond. Single bonds allow for free rotation of atoms around this bond, whereas if you have a double bond, that is to say 2 pairs of electrons being shared by 2 atoms, you don't get that kind of free rotation anymore. It makes for a very rigid bond. Down here with water, we have this polar covalent bond. Oxygen is more electronegative than hydrogen, so the electrons will spend more time around oxygen, making it like partially negative, and conversely, the hydrogens that have the electrons for less time, they become partially positive. But they're not full charges by any means. Here are some functional groups and linkages. I won't be testing you on the actual structure here, but you should be familiar enough with them to recognize them on site, and I'll probably refer to them by name whenever we go throughout this course. I'll say like, oh, there's a hydroxyl group here, or an amino group there, or a phosphate. Phosphates are really important. Look at all those negative charges. We'll talk about pyrophosphates really briefly, esters, carboxyls. Carboxyl for sure. But yeah, mostly in this course, hydroxyl, amino, phosphate, carboxyl. Those are the 4 big things that we'll talk about. Amide linkages come up, but I don't really talk about them. Okay, we've got our non-covalent interactions. Again, here electrons are not shared. So you could have something like a hydrophobic interaction, right? Here in the center, these 3 molecules are nonpolar. They want to interact with each other. They don't want to interact with these polar water molecules. You could also have hydrophilic interactions, right? In an ionic bond, you have those full charges attracting. So in this molecule of salt, sodium is positively charged, chloride is negatively charged. They're binding to each other via ionic bonds. You could have partial charges where you have, again, that partial negative on the oxygen attracted to the partial positive on this hydrogen. They're going to want to interact. And so here you have hydrogen bonds, hydrogen interacting with oxygen, so to speak. Okay. I like this figure because it shows you a visual representation of what might be happening as 2 proteins are interacting with each other. It shows you how it's really the combination of all different kinds of bonds, ionic bonds, hydrogen bonds, hydrophobic, these non-covalent, non-polar interactions rather, that come together at the surface to make for a stable complex between protein A and protein B. Let's say we keep protein A the same, we switch protein B for protein C. All of a sudden, protein C doesn't have those positive charges here that protein B did. Now instead we have negative, and negative repels negative. It still has hydrogen here, but it's not a hydrogen involved in a polar bond. It's a hydrogen involved in a like a nonpolar bond, and so we're not getting the hydrogen bonding that we did there. Again, we have these same charges here that are repelling. We still have this one hydrogen bond here in the middle and here at the top, but again, they're very different proteins. It's not going to be a stable complex like you got with protein A and protein B. Um, in this case, the binding is 100% reversible, right? You need the molecules to be able to dissociate, to be able to bind, and then to unbind. Okay. Another cool thing is that biomolecules are actually malleable. Okay. And so as one protein comes up to another, they can almost finagle its binding partner and induce a change in shape, okay, such that we can line up these interactions more tightly. And we say when this happens, this is an induced fit. Okay, but let's talk about carbohydrates. Okay, these are all of your sugars. These are mostly carbon and water. You need carbohydrates in your nucleic acids. You have that sugar phosphate backbone. They're going to give a lot of strength to your structural components. They're what store a lot of your energy. And we generally categorize carbs into either simple, whether like a monosaccharide, one ring, or disaccharide, two rings, or a very complex in which you have lots of these rings joined together, a polysaccharide. And we say that they're joined by glycosidic bonds. Okay. So again, here's your simple carbs. These are what we call our sweet saccharides because they taste sweet, right? If you can eat it. You're going to classify them based on the number of carbons. They're really stable in this ring structure. And we can pull out a water right here to make a disaccharide when we join two monosaccharides together. This is what we call dehydration synthesis. But for the purposes of this course, right, just kind of know generally monosaccharide looks like this, disaccharide looks like that. If we start pulling out water, they join together. Polysaccharides, we use them for fuel, we use them for structure, right? So you have your starches that have all of these monosaccharides joined together. They can even start to make these branching structures. But we have polysaccharides all throughout your extracellular matrix, polysaccharides that make up the cell wall in plant cells, polysaccharides that are attached to your lipids and attached to your proteins and convey all kinds of signaling functions. But we'll talk more about that in unit 3, maybe 4. I forget. All right, so that's our carbs. Then we have our lipids. Lipids are just your carbon and hydrogen. Mostly lipids are nonpolar. They don't dissolve in water. You have lipids in your membranes. A lot of your hormones are also considered lipids. Like your carbs, they store energy. And we're going to classify them as either a simple lipid, a complex lipid, or a steroid. So first off, we have our simple lipids. So we have our fatty acids, right? You have this carbonyl group right at the end here. You have this long hydrocarbon chain. Again, just carbons and hydrogens, okay? And these are all different based on the number of carbons that they have and even sometimes the kind of linkage. So see here, stearate and oleate both have 18 carbons in their carbon tail, but they're different in their structure because oleate has this double bond in the middle right there. And once we have our double bond, right, that this particular bond locks in place, it's rigid, you don't get that free rotation. And so if you saw this in your lipid membrane, you'd say that there's a kink in that lipid tail. You also have your triacylglycerides, okay, tri because you have 3 of these tails, acyl because they're connected here to these, I should know my biochemistry a little bit better, but I'm sure that has to do with the chemistry there. You have your glycerol, right, this is your structure in the red box. So 3 fatty acids coming off of your glycerol. And this is also considered a simple lipid. Your complex lipids are going to have more kinds of elements. They could have phosphorus, they could have nitrogen, they could have sulfur. This is your basic building block of any kind of membrane, is this phospholipid. We'll talk more about different kinds of lipids that are found in your membrane and even different subcategories of phospholipids once we get to the last unit. But suffice to say, we have this phosphate in the head group and some other strange thing off the end here. R just kind of a placeholder umbrella thingamajig. Again, we have our glycerol, but now here we only have 2 of these fatty acid tails. And so all the polar heads are going to interact with each other. All the non-polar tails, fatty acid tails, are going to interact with each other on the center here. We'll have our polar head face the outside aqueous polar environment. thus protecting our nonpolar tails at the center there. We'll talk more about these kinds of structures later on in this course. Finally, we have our steroids. Okay, steroids are still considered lipids. Most of them have these fused carbon rings where they're not kind of standalone, but they're going to be sharing at least 2 carbons with the neighboring ring. You'll find them in cell membranes to give additional structural integrity. They'll be used as hormones. And I want to point out here, sterols are a kind of steroid that has a hydroxyl group. So again, this guy down here, the O and the H. We'll talk about these guys once we get into our cell membrane lecture, again, later on in the course. Okay, so that's our lipids. Moving on, we have our proteins. Okay, and so the monomeric building block of a protein is your amino acid, where you have the central alpha carbon that is connected to a hydrogen, a side chain R group, and you have an amino group on one side that we call the N-terminus because you have a nitrogen in there, and a carboxyl group on the other hand called a C-terminus because we have a carbon in there. Okay, and we're going to link our amino acids together to make our proteins. We link by connecting carboxyl groups to our amino groups. We pull out a hydrogen, much like, water, just like you saw with monosaccharide synthesis into disaccharides. You're going to make this peptide bond, right, with this carbonyl and the nitrogen there. Proteins are always numbered from the N-terminus, from the endmost amino acid to the C-terminus of the other endmost amino acid. So here, right, you have the N-terminus here, so we say that this is the first amino acid, second amino acid, third amino acid, ending with your C-terminus or carbonyl group that's exposed. Okay, so the N-terminus has an amino group that's exposed, the C-terminus has a carbonyl group that's exposed, and the N-terminus is always first. Okay. This slide is so that you can see all of your amino acids in the same place. I don't need you to know the structure. Okay, that's what hopefully that you will memorize in your biochemistry course. But I do want you to know a few things about them. One is you should generally know whether any particular amino acid is hydrophobic or hydrophilic. Second, you should know which amino acids are charged, and of those charged amino acids, which ones are basic and which ones are acidic. There's only 3 basic ones and 2 acidic ones. Third, you should be familiar with their 3-letter and 1-letter abbreviations. Okay, and fourth, you should know how we name amino acids within protein structure. So they'll use the 1-letter code for the amino acid, so K for lysine, and then a number to indicate what position it is within that polypeptide chain. So K48 means lysine at position 48. But right, as is such the case, the mutations happen, and sometimes that native amino acid gets changed to something else. We'll then follow this number with the amino acid that it got changed to. So in this case, we have K48R, which means that this lysine at position 48 was mutated to arginine. The other thing I want you to know here is that some of these amino acids can be modified by adding different kinds of functional groups, like for, say, a phosphate group. So we'll be talking about phosphorylation of different proteins, and these are the only ones that can be phosphorylated. You have your tyrosine, serine, threonine. You have other amino acids that can be modified on their histone tails, like lysine or arginine. You have amino acids that can be ubiquitinated, that's just lysine. You have cysteine that functions in disulfide bonds. These are the 4 main kinds of protein modifications we'll be talking about during this course. Okay, but again, you don't need to know the structure. I'm never going to ask you to draw that out, but I do want you to be familiar with all of these characteristics associated with amino acids. Protein structure has 4 levels. That first level is just that primary protein sequence, which amino acid is joined to the next, what order does it occur in. Okay, that's the sequence. Second, what happens when those amino acids form hydrogen bonds with each other, right? How does the protein locally fold into these alpha helices or beta sheets? Third, how do these alpha helices and beta sheets then kind of globularize, that's a made up word, doesn't exist, don't look it up, the protein into a more compact structure, right? How do the alpha helix here interact with another alpha helix or with a beta sheet? And finally, a quaternary protein structure, not every protein has this, just some. How does that protein that we've just folded interact with another protein? It could be the same protein, it could be a different protein, right? But that's your quaternary protein structure. So a little bit here about alpha helices and beta sheets. In alpha helices, you'll see the hydrogen bonds being formed between not neighboring amino acids, but it's about every fourth one. I think it's, okay, I don't know the number off the top of my head, but that's something that you should have learned in fundamentals. So you see we have the amino group here interacting with the carbonyl group of this other amino acid, and we have that same kind of hydrogen bond going on over and over again, and you're just pulling it tight. Notice here that all of the R groups are pointed outwards from the alpha helix, okay? They're not pointed inwards, there's not enough room. So yeah, they have these R groups basically being perpendicular to the alpha helix. And those side chains will let you know whether the alpha helix is hydrophobic or hydrophilic. And again, these R groups are going to be interacting with the environment. You have this other secondary structure, these beta sheets. Okay, so what's going on here? Again, you have the amino groups and the carbonyls forming hydrogen bonds with each other, but you almost have this folded structure going on. And so what's happening here? Again, you can see all of these hydrogen bonds happening to have one strand interact with the next strand over, but you have this folded action going on between the amino acids. Strands could be anti-parallel, in which they're going in the opposite direction, the individual strands, or they could be parallel, they're all going in the same direction. It kind of just depends. Notice here that the R groups are pointed above and below this plane of the hydrogen bonds. Okay. Another term I want you to be familiar with are these structural motifs. Okay. This is when you get at least two secondary structures that make a distinct 3D structure, and we're also where you get this motif appearing over and over again in different proteins. A lot of the time, these motifs will perform a specific function. So for example, this coiled coil motif is usually a protein interaction motif. See how you have all of these hydrophobic amino acids along the interior of this coil of this interaction surface, allowing these two strands to wind tightly around each other. We'll talk a little bit about helix loop helixes and zinc fingers when we get to transcription factors later this unit. Then we have our protein domains. Okay, so I'll use this term throughout this course, and we use this to mean whenever a section of that polypeptide is self-stabilizing, it folds independently. And so typically in any given protein, you'll actually have not just one, but several domains. What's also really cool is that you can pop these domains in and out of different proteins. And so you start to get really cool recombination things. We'll talk all about this in the genome lecture. But if you see the same domain appearing in different proteins, you can usually assume that it's going to perform the same function. So an ATPase domain is something that's going to hydrolyze ATP and channel that chemical energy and turn it into mechanical energy to perform some kind of movement. You might get any number of these different secondary structures, alpha helices, beta sheets, random coils, and usually the domains are separated from each other by linkers, these kind of disordered regions that are much more flexible. So you have the blue up here is one domain, the white down here is another domain, the green down here is a third domain. You'll see that they're linked more loosely, not in the stiffer secondary structure. Domains get defined usually by what kinds of reactions they can catalyze. So you could say kinase domain is going to do a lot of phosphorylation. Um, ion helicase domain is going to unwind DNA. A nuclease domain is going to cut DNA or RNA, right? Or by binding ability, what kinds of things do they recognize? So an SH2 domain is going to recognize phosphotyrosines and so on and so forth. Okay. But just so you know, right, not all of your amino acids are going to fall into these secondary and tertiary structures, and we call these unfolded regions intrinsically disordered regions. Okay. Okay. Just so you know, proteins can have all kinds of things stuck on them, and these are going to be covalent attachments. You can have glycoproteins that have sugars, nucleoproteins with nucleic acids. Well, maybe not your nucleoproteins or your metalloproteins, but typically your lipoproteins, your phosphoproteins, your glycoproteins, those are covalent attachments. Nucleoproteins and metalloproteins are non-covalent interactions. Okay? But yeah, you'll see all kinds of fun combinations of things. Okay, finally, okay, you thought this lecture was never going to end. I also thought so too, but it is ending. Here we go. You have your nucleic acids. So our two main kinds of nucleic acids are DNA and RNA. Okay, so DNA is your genetic blueprint. You got all the information to build your cell. RNA does everything else, right? RNA you see a lot in protein synthesis, whether it's by carrying the information from DNA to the ribosomes, or by being part of the ribosome itself, or by bringing the amino acid to the ribosome to actually translate that nucleic acid information into proteins. Fun stuff. We'll talk about that in translation. RNA has no hereditary function in cells. If you want to learn more about how RNA can be hereditary in viruses, come take virology with me. And you can have structural roles, regulatory roles, all kinds of stuff. Your general components of a nucleic acid are the backbone. So you have your sugars, right? Whether it's ribose with this extra hydroxyl group for your RNA, or deoxyribose for your DNA. Phosphates that link, right, the sugars together, and importantly, your nitrogenous bases. Okay, whether it's your purines. Okay, so you can remember pure as gold, adenine, guanine. Or your pyrimidines, which are cytosine, thymine for DNA, and cytosine and uracil for RNA. Okay, and you can remember which ones have an extra ring by the mnemonic pure as gold, and also girls have a purity ring. It's kind of dated, I know, but it makes sense, so I'm telling you guys this. So your adenines and guanines that are purines have the extra ring. Okay. Your nucleosides have bases and sugars only, okay? Whereas a nucleotide is going to have your base, your sugar, and at least one phosphate up to 3 phosphates. So that's a small distinction there. Know also that your nitrogenous bases are making these hydrogen bonds, and they're bonding specifically between purine and pyrimidine pairs. So adenine bonds with thymine, okay? Guanine binds with cytosine, okay? And in RNA, adenine bonds with uracil. See how uracil and thymine are very, very similar, except for the fact that thymine has that additional methyl group right there. The other really important thing you have to know is this numbering of nucleotides. And this numbering comes from the fact that we number the carbons 1 through 5, 1 prime, attached to the nitrogenous base, and then going around away from the oxygen, 2, 3, 4, 5. And the linkages that happen between each base happen at the 3 prime and the 5 prime carbon. And because we grow nucleotides by nucleic acids rather by adding nucleotides to the 3 prime end, right? We say that nucleotides have a directionality where they're linked 5 prime to 3 prime, and by default, if there's no numbers there, you assume that the numbering sequence is from 5 prime to 3 prime. Okay. The 5 prime end of the mRNA also is going to correspond to the N terminus of a protein, whereas the 3' end of messenger RNA corresponds to the C terminus of the protein, so the beginning and the end, right? We write the beginning at first and the end one last. So 5' to 3'. DNA strands are going to wind together. We make this anti-parallel double helix. Again, the strands are held together by your hydrogen bonds between your nitrogenous bases. Okay, and again, you see you have 5' to 3' in one direction. Okay, and the other strand is going in the other direction, where the 5' is at the bottom, 3' at the top. Okay. RNA typically exists on a single strand, but given that it's a single strand, it can base pair with itself, and in so doing, make these secondary structures. These are different kinds of secondary structures than your protein secondary structures. We call a secondary structure in RNA just any area where there's base pairing, such that it's stiffer, it's not as flexible. You can't just have it float out into the abyss like it's constrained. It can't just do anything that it wants. Okay, here's just a final little summary slide with all the macromolecules that we talked about. Your proteins, right up here, your amino acids that link together to make polypeptides, your nucleic acids with your nucleotide. You can tell it's a nucleotide because you have your phosphate that links together to make your nucleic acids, your saccharides that make polysaccharides, and then finally we have our lipids down here. And here we're only showing you our phospholipids. So I hope this was helpful. Yeah, I will see you guys in class.